Now, we just need to understand that concept of linked genes to help us move on to the major topic of this vodcast, which is recombination. So, on to the main idea here. Now that you understand linked genes, we can talk about when those linked genes split up. And the process of splitting up linked genes is called crossing over. It's actually a really nice name for it because this is what actually happens. The chromosomes actually cross over. We'll talk about this real shortly here. So this process of crossing over occurs during prophase 1, so this is in meiosis 1, when the homologous chromosomes form the tetrads. In other words, when they line up right across from each other. Okay, so remember I was diagramming meiosis 1 and I showed how those homologous chromosomes have to line up next to each other. This is when this process of crossing over happens. And as I said, the process is exactly what it sounds like. Here we have our two homologous chromosomes lined up across from each other. Perhaps the white one is from the mother and the blue one is from the father. They've already formed their sister chromatids, so they're ready to go through meiosis. Those two chromosomes that are closest to each other, lined up across from each other, actually stick to each other and cross over occasionally. Okay, chromosomes are made out of DNA, which we've talked about, and also some other proteins that help the DNA kind of wind up and stick in the right shape. And the chromosomes can be kind of sticky. In other words, there are charges on the chromosomes that attract to each other and stick to each other. So we have actual entanglement or wrapping of the chromosomes that are closest to each other in this tetrad. Then, when they pull apart, later in meiosis 1, during anaphase 1, those parts that were crossed over actually swap spots. So now on this set of chromosomes in this homologous pair, we've got a little bit of the dad's chromosome is stuck onto one of the mom's, and a little bit of the mom's chromosome is stuck onto one of the dad's. And you can see, hopefully, that those are different alleles. Okay, the mom used to have two little c alleles. Now she has a little c and a big C. The dad used to have two big C alleles. Now he's got a little c and a big C. So now those genes, the versions of the genes, the alleles that were carried on those chromosomes are different, which means the combinations in the gametes, when meiosis is all done, is also going to be different. We'll diagram that in a little bit. But before we move to that, I just want to let you know that in crossing over in this process, it's more likely to happen between genes that are far apart. And what do I mean by that? If you look at this chromosome again, look at these A genes and B genes way up here on the top of the chromosome. You see how they are spaced very close together? Okay, so if I was to redraw one of these chromosomes, be like the A gene is right here. Let's make that a little darker. Let's make it in blue. There we go. The A gene is right there, and the B gene is right next to it. So they're located very close on the chromosome, whereas the C gene is way down here. Okay, if we try to physically take those two chromosomes and cross them over in this little area here between the A and the B genes, it'd be very difficult to physically cross them over there, right? There's just not as much space. But there's a lot of space between the B and the C chromosomes. Okay, a lot of space between those, which means it's easy for there to be a crossover between those two genes. Okay, so crossovers occur more frequently between those genes that are farther apart on the same chromosome. So the whole point of getting into this process of crossing over is that it can create new possible gametes. And we call those new possible gametes the recombinants. It comes from the word recombine. Okay, so we're recombining the different alleles so that different possible gametes can be formed. Because we just talked about linked genes and how if genes are on the same chromosome, it limits the types of gametes that can be made. However, if the genes are allowed to cross over, okay, like these did here, we get some new gametes that wouldn't have normally been made. And again, those gametes are called recombinants. All right, back to some diagrams now to look at what's actually going on with these recombinants. Now this diagram should look familiar. Okay, we just looked at this. We've got a particular cell here, okay, that has linked genes. So we've got one set of chromosomes. The genes A and B are linked, so they're on the same chromosome. And remember, the outcome was two possible gamete types. Right? We just have the completely dominant gametes and the completely recessive gametes. And this is how most of your chromosomes would end up sorting out because, as I said, you contain many genes on one chromosome. So a lot of those genes are inherited together on the same chromosome and can't be separated. Except if we have crossing over. Again, crossing over adds in that chance of mixing up those alleles a little bit in the gametes, in the egg and the sperm cells. So let's take a look at how that happens. Instead of the two chromosomes, I'm just backing up here again to remind you what this looks like. Instead of the two chromosomes staying as big A with big B and little a with little b, if we have a crossover, getting there, those chromosomes actually intertwine with each other, they get tangled up with each other, and separate a piece out from each chromosome. So we're going to highlight our little bits of chromosomes here. We'll make the first side pink again. So there's chromosome on the left side, and then the other side is going to be yellow. Okay. Notice that the pink chromosome originally had a big A and a big B, and the yellow chromosome originally had a little a and a little b. So we're going to diagram that out. They've lined up in metaphase 1, so they're right across from each other. Remember, they cross over, so I'm going to draw them overlapping so that when they separate out into two cells, a piece of each chromosome actually rips off and sticks to the opposite chromosome. So now we're going to have our original X chromatid with the big A and the big B on it, and then the sister chromatid is going to have a little bit of the other chromosome on it. And on the other side, we're going to have the original chromosome with the little a and the little b on it, and then the sister chromatid is going to have a little bit of the other chromosome on it. Okay, so they swapped sections, and on that section was a gene. Okay, in this case, it was the B gene that got swapped. Now we're going to do the same thing, go through meiosis 2 as we did before. Okay, separate them out. Divide those sister chromatids. I have 4 possible gametes. Okay, on one side we've got all yellow. On the other side we've got a little yellow and a little pink. Same thing on the second set of cells. One side we've got pink and a little yellow, and then on the other side we've got all pink. And now remember what traveled with those chromosomes. The yellow one, remember, was originally little a, little b. Okay? This yellow one, we still have the little a, but now we've swapped it for a big B for the bottom. The pink chromosome was originally big A, big B. This other cell now has a big A with it, but attached to it or swapped out was now a little b from the yellow chromosome. So now we've got 4 different possible gametes, right? We've got the big A, big B, which I'm showing here, big A, little b, which I'm showing here, little a, big B, which I'm showing here, and little a, little b, which I'm showing here. The ones that came from the crossing over, okay, the alleles that got swapped are this gamete and this gamete, right? Because these are both half pink and half yellow. And that corresponds over here to this gamete and this gamete, okay, this egg or sperm cell. Those gametes that are the result of crossing over, remember, we call the recombinants. Okay, so those ones that I highlighted in green are the recombinant sperm or egg cells. Those are the ones that happened because of crossing over. If no crossing over occurred, we would only get these gametes, because that's how the genes are attached on the same chromosome. So if you're drawing this diagram, you want to make sure that you include the fact that those two on the right-hand side are your recombinant gametes, the ones that are the result of crossing over. Okay, so why is it important to understand, number one, that genes are linked, and number two, that linked genes can cross over to form these different versions, these recombinant gametes? Well, we've been talking about all along how we get variation in our offspring. Okay, why is it that you look different from your brother or your sister? Or not all humans look the same even though we share a ton of DNA? Well, we've already discussed two versions up till today. So we talked about meiosis. Remember how those homologous pairs line up in meiosis can affect what genes go into the gametes, which versions of the genes, which alleles. Okay, so it's a random shuffling of the deck. That's the law of independent assortment. We also discussed before today the process of fertilization, how one egg and one sperm coming together can be different depending on which combinations you have, and that's what the Punnett squares are all about, right? What is the probability of this genotype happening if this egg and sperm get together versus another genotype happening if this egg and sperm get together? Right? So meiosis and fertilization are two ways that you can get variation or differences in genetic combinations in the offspring. Now, today's podcast gives us the third way that variation can happen in offspring, and that is through recombination or crossing over. If linked genes cross over and swap their alleles, you're going to get different outcomes in the offspring, which were the result of the different outcomes in the gametes. So these are the main three ways that you get the difference between siblings. Okay, I don't look like my brother because the egg that made me was lined up differently in its chromosomes than the egg that made my brother. The sperm that fertilized my egg was different than the sperm that fertilized my brother's egg. Also, I may have had some recombination events in the formation of the gametes that ended up being me that my brother did or didn't have. So all of these possibilities contribute to the variation or the differences we see in people in the same family.